Assalamualaikum and welcome back to our practical class. So today is going to be your last practical, which is practical 5 for this last semester, which is semester 2 in session 2020 and 2021. So for today's topic, you are going to study about the respiratory structure, gills of the bony fish. As you already learned in your previous lecture, alright, so the oxygen and carbon dioxide dissolve in water, and most fishes exchange the dissolved oxygens and the carbon dioxide in the water by means of the gills. And the gills lie behind and to the side of the mouth cavity and consist of fleshy filaments supported by the gills arch and filled with the blood vessel, which give gills a bright red color. And water taken in continuously through the mouth passes backward between the gills and between over the gills filament, where the exchange of gases take place. The majority of bony fishes species have 5 pairs of gills, although a few have lost some over the course of evolutions. And the functions of operculum, it can be important in adjusting the pressure of water inside of the pharynx to allow the proper ventilations of the gills. And the functions of bony fish gills, which is to extract the dissolved oxygens and excrete the carbon dioxide in the seawater. And the filament of the gill are covered with a thin epidermal membrane that folded repeatedly in the plate-like lamella. And the lamella has rich supply of blood vessels. The gases exchanged are aided by the countercurrent mechanisms. To maximize the gas exchange, the gills are adapted to the fish body shape and have a large surface of area. And this process is aided by the muscle movements, mouth, operculum, and the closed circulatory systems. So for today's practicals, our objective, we have 2, which is the first one is to observe the position of the gills, the mechanism of respiratory processes, and the detailed structure of the gills. And the second objective for today's practical is to observe the morphology and structure of the fish. For the materials that we are going to use for today's practical, okay, the first one we have bony fish, which is selah kuning. We also have the dissecting set, okay. We also have the distilled water, scissors. This is the compound microscope and the dissecting microscope. Alright, first method, observe the morphological structures of the fish. Draw and label the structures such as head, body tail, fins, which is we have dorsal fin, pectoral fin, pelvic and anal. We also need to draw the eyes, mouth, operculum and lateral line. Observe the anterior part of the fish, particularly the coordinations between the mouth, gills and operculum. For the third method, you need to remove the whole gill structure, and you take only one piece and put it in the petri dish that filled with distilled water. Change the distilled water until there are no traces of blood or tissue. Then you need to observe under a dissecting microscope the gills recurse, gills arch, and gill filaments. Draw and label those structures. Then for the last method, pick up one gill filament using a forceps. Then with a pair of scissors, cut approximately 5 milliliter length and put it in a drop of distilled water on the glass slide. Gently cover the filament with a cover slip. Then you need to observe, draw and label the structure of the gill filament. Guys, so we have come to the result and discussion part. For the result, you need to prepare a report with 4 label diagrams, which is the first one. You need to draw the general morphological structures of the fish, such as head, body, tail, fins that comprise of dorsal, pectoral, pelvic and anal, eyes, mouth, operculum and lateral line. Then for the second diagram, you need to draw and label the exposed gills. For the third diagram, we need to draw the observed diagram under a dissecting microscope with the gills recursus, gills arch and gills filament. Draw and label those structures. And lastly, for the fourth diagram, you need to draw and label the gill filament. Then for the discussion part, you need to discuss the respiratory mechanisms of the fish gills. Then you need to compare the gills of the bony fish and the gills of the cartilaginous fish. That's all. See you guys. Thank you.